be a keynote speaker, and I hope I don't disappoint you too much. Right. I need to disclose at the beginning that I do receive some royalties from Sangamo, and I'm on the scientific advisory board for Genetics. And later in my talk, I'm going to be mentioning some things that are happening in both of those companies. So if you think I'm just trying to enhance the value of those companies, you can disbelieve that if you want to. So this is an outline, brief outline of my talk. Because I'm the oldest person at the meeting, I'm allowed, and in fact, mandated to do some history. There's a little bit of the history and some of the middle part, I think all of the middle part is research from my own lab, but quite a bit of what I'm going to be talking about is a little bit from the journal. For me, the issue of the development of the targetable nucleases began with thinking about gene targeting. And I think people are aware that in mouse, there was a gene targeting approach that was available, developed by Larry Pecky, where even though the targeting frequency when you put in a homologous donor DNA was very low, Pecky had developed selection procedures to allow you to find the very rare cells, particularly embryonic stem cells, that carry the modification. But we've known for kind of a long time that frequency of homologous replacement goes up dramatically, many orders of magnitude, if there's a break in the target at the time of the introduction of this donor DNA. And so the problem of enhancing gene targeting is reduced to one of making targeted double-strand breaks. And so for me, that was where things began. The first of these targetable nuclease reagents is Zinc nucleases, and I'm going to just go through a little bit of the history, making the point that the ZFN technology didn't arise de novo out of the primordial soup or somebody's brain. That actually came from the confluence of different research findings, one of which was this discovery that double-strand breaks stimulate homologous recombination as well as their zinc fingers or modular DNA recognition domains found in natural transcription factors, and then studies on the FOAT1 restriction endonuclease that showed you could separate the recognition and cleavage domains very effectively. This is just a little bit of the data that led to the appreciation of double-strand breaks stimulate homologous recombination. It was known for a long time that radiation damage stimulated recombination. I don't know how well you can see this, but these are Harlequin chromosomes. I think they're actually from a patient with Bloom syndrome or something like that that enhances this sister chromatid exchange. But upon radiation damage, you enhance this homologous exchange between sister chromatid. Meiotic recombination in essentially all organisms is initiated by intentional double-strand breaks that are made by the SCO11 protein, other organisms. And then there were model experiments that were done in yeast, initially by Jim Hager's lab and Maria Jason's lab, pioneered these experiments in mammalian cells using homing endonucleases or meganucleases with very long recognition sequences, and showing that if you introduce the recognition sequence for these enzymes into novel chromosomal locations, and then introduce the enzyme that you can stimulate both targeted mutagenesis and targeted homologous recombination. Zinc fingers were discovered essentially informatically initially by Aaron Klug's lab, and they did some experiments that demonstrated that they were zinc-binding proteins. But it was really the crystal structure of a three-finger domain bound to DNA, which was done by Nikola Pavlovich and Carl Pagel's lab, that got people excited about these recognition elements because they're quite modular. Each finger has a stereotyped three-dimensional structure, and individual fingers recognize individual triplets or quadruplets in their target DNA through binding of side chains on 3 or 4 residues within DNA sequences, with different amino acids in these positions. And then if you look up FOLK1 in the New England Biolabs catalog, it says that it recognizes an asymmetric 5-base sequence that's 9 and 13 bases away from strands. And that's because the recognition and cleavage domains of this enzyme are naturally separable. And Chandra-Segaran's group demonstrated that by limited proteolysis, they could isolate all of the DNA binding activity in this 41 kilodalton long fragment, sequence-specific DNA binding, and non-specific DNA cleavage activity was in this 25 kilodalton fragment. And Chandra went on to begin linking the FOLK1 cleavage domain to different DNA recognition domains. You may not know that the first fusion he made was to a Drosophila homeobox recognition domain, not to zinc fingers. But when he did make fusions to zinc fingers, my lab got quite excited and initiated a collaboration with him. Because of our knowledge of A, that double-strand breaks stimulate mutagenesis and recombination, and B, that zinc fingers are flexible DNA recognition modules. So we had a collaboration over several years with Chandra's group demonstrating that, like the natural FOLK1 cleavage domain has to dimerize to cut DNA. The dimer interface is weak, so you need to make zinc fingers that bind two sites next to each other. We did some experiments looking at the relationship between the spacer on the DNA and a linker protein. We demonstrated that cleavage and recombination are possible in a chromatized target using xenopus oocytes and synthetic targets for that. And at the time we were doing these experiments, we knew that there was zinc fingers available for many of the 64 possible DNA triplets. So it seemed plausible that we could begin to target arbitrary DNA sequences making variants on these fusions. And one of the key features of the ZFMs is that it has separable recognition and cleavage domains so that you can manipulate one, modify the recognition specificity without affecting cleavage. And people have also manipulated the cleavage domain without any consequences for DNA recognition. And the first genuine genomic target that we ever hit with the zinc finger nucleases was within the yellow gene of Drosophila. And over beer or something stronger, I can tell you a long, boring story of the history of this. But suffice it to say that we made novel combinations of zinc fingers to recognize sequences in the yellow gene. Each of these combinations was linked to a monomer of the FOLK1 cleavage domain, and when both were expressed in flies, we got mutations at this site within the yellow gene, and later demonstrated that when a donor DNA was also provided, that we could get homologous repair of the cut site in the yellow. Now, I went through that story because the elements that came together to lead to the use of zinc finger nucleases came from rather disparate places. And the same thing is true of the more recent entries into this nuclease field. The TALENS arose from studies on bacteria that infect plants, and these bacteria was various Xanthomonas species and related genera. Amongst other things they do during infection, they generate proteins that are secreted into the host cell, go to the nucleus, and regulate host cell genes. So they're bacteria generating transcription factors that will act in the host cell, in the plant cell. And when people made a correlation between the individual repeated domains in the DNA binding domain of these bacterial proteins and the target sequences in the promoters that they were regulating, they discovered there was a code. It's illustrated most clearly down here that frequently when a seed was present in the recognized strand of the target, the TAL subunit had histidine and aspartic acid at key positions within the 34 amino acid repeat. And so this code led to the generation of analogs of the zinc finger nucleases, where the zinc finger domain was replaced by these tail DNA recognition domains. And for a number of reasons, these were quickly adopted by a number of people. For one thing, there is a single module for each base pair of DNA. You don't have to try to find a module that recognizes a particular triplet. There's one defined module for each base pair. So there's a robust code that's very easy to design. The other thing I think that led to the rapid adoption is that the orientation of the DNA binding domain on the target DNA was N to C terminal relative to 5' and 3' orientation of the target. And the zinc finger nucleases confused a lot of people because the recognition was backwards. The N-terminal finger recognizes the 3' end of the recognition site, and people just didn't want to have to deal with the backwards recognition. So TALENS were rapidly adopted, and again, they have separable DNA recognition and cleavage domains, which makes them easy to manipulate. Several people have put up slides that look like this, describing the origin of the CRISPR Cas system, where bacteria, one of their modes of generating immunity against invading viruses and plasmids is to capture sequences from previous invasions and then use them to generate RNAs that will direct cleavage in the next invasion by this Cas9 protein. And this led to the production of synthetic versions of this Cas9 protein, the guide RNA that is all in one molecule. But again, the recognition by RNA, and a little bit by Cas9, and the nuclease functions are separable so that you can manipulate one without manipulating the other. And because here recognition is provided by a Watson-Crick base pair, and there's only one constant protein that's involved in generating cleavage, people have adopted this, as we have seen, in very exciting ways, very, very rapidly. So, the point of going through that is to say that in none of these cases was the research that led to these novel cleavage reagents targeted at that outcome. It was research broadly in different arenas that led to the ultimate utilization of these components for genome engineering. All right. One of the things that we were quite interested in my lab was how we could manipulate the system and to favor the outcomes that we or others might want. And one of the starting point for this was that when a double-strand break occurs in an endogenous chromosome, cells will go to great lengths to repair that break so that they don't lose a piece of chromosome. And normally, homologous repair would occur off the sister chromatid, but there's an alternative pathway that people have talked about called non-homologous end joining, where ends are just slammed back together without any concern for homology. This generates localized mutations, and you can fool cells into using a template for repair, a homologous template for repair that you've provided, instead of their using their own sister chromatid. But the point is that all of these reagents only make a double-strand break in the DNA. They don't direct how repair is going to occur, except to a very limited extent. So understanding how repair activities in different organisms and different cells seem to be important to being able to control the outcomes. And for these experiments, again, we're working in Drosophila. Our best zinc finger nucleases, and we've also used TALENs and all the CRISPRs, as I'll tell you a little bit about later. Our best zinc finger nucleases were for another Drosophila gene called ROSI. We made ZFNs that bind this target sequence and provided a donor DNA that had the recognition sites that we did to replace by in-frame stop codons and a diagnostic restriction site. It's very easy to recognize flies that are mutant for ROSI. Wild-type flies have a bright red eye, and rosy mutants have a duller red eye. And even the PI in the lab is allowed to screen for new mutants because it's so easy to do. So these are some of the issues that we addressed using rosy as our test system. So once a cut has been made, there can be non-homologous end joining to repair the break, and this will often generate new rosy mutants for cleavage at that site in the rosy mutant. But if we provide a donor DNA that also will generate new rosy mutants, homologous recombination can generate rosy mutants that we can score, and we can distinguish these two. So we asked how much homology is required in the donor in order to be successful with homologous recombination. I will show you those data. But we were also quite interested in how could we influence the balance between end joining and homologous repair. And another issue, which I think is very important, is how far from the break site will sequences from the donor be incorporated into the target. And I'm going to show you a little bit of data about both of those issues. So it's true in most systems that people have dealt with, not true in yeast, but most other eukaryotic systems, that a simple break is more often repaired by end joining than by homologous recombination. And that was true with cleavage at the rosy locus in Drosophila as well. Somewhat less than 20% of all the new rosy mutants arose by homologous recombination, and the remaining 80+% were end joining mutants. But when we redid the experiment in flies that were defective in one of the components of the canonical end join system, DNA ligase 4, the proportion of homologous recombinants that were amongst the new rosy mutants rose dramatically. We had some experiments where essentially all of the new mutants were the product of homologous recombination. And so that's great. You knock out ligase 4, you get more homologous recombination without losing much of the overall mutagenesis. Flies can get along without DNA ligase 4, but many organisms cannot comfortably. So using mutations in ligase 4 is not a good strategy generally for improving homologous recombination. If there were small molecule inhibitors that were very specific for DNA ligase 4 and you can use them transiently, that would be a good way to try to accomplish the same thing. But reducing the canonical end joining pathway leads to an increase in homologous recombination. That's good. This other issue that we are quite concerned about is conversion tracts. So at Rosie, we were fortunate that the strain we were targeting had a chromosomal sequence that differed somewhat from the donor we were introducing for homologous recombination. And so we could distinguish chromosomal from donor sequences by restriction enzyme cleavage, or we used alternative approaches to this more recently. And in Drosophila embryos, where we're doing the cleavage and whatever repair by embryo injection of the nuclease and the donor DNA, we found that the conversion tracts were very broad, regardless of whether we were wild-type embryos or ligase-4 mutant embryos. And what that means is, if you've gone to a lot of trouble to develop a good cleavage reagent for a particular site in a gene in Drosophila, you can use different donor DNAs to introduce novel sequences over a very long range away from that cleavage tract. You don't have to develop new cleavage reagents. You use the same different donor DNAs. And we've extended this out on this side more than 3 kb and still get roughly 50% conversion over quite a long range. Unfortunately, in mammalian cells in culture where people have studied this, these conversion tracts are very narrow. So you don't have this opportunity to use multiple donors over a very long region to introduce a range of mutations. So it would be wonderful to know what it is that makes the difference between these two situations, what limits the conversion tracts in mammals, or what enhances the length of the conversion tracts in Drosophila embryos. And just in passing, I can say that when we do this experiment in Drosophila larvae rather than in embryos, we get sort of an intermediate answer, that the conversion tracts are broader than in mammals, but narrower than what we see in embryos. So there are features of the repair machinery in any cell type that are influencing what the outcome is going to be. We were interested in comparing aspects of the outcome, efficiency, consequences, using the different platforms. And so I've talked mostly about things that we've done with CFNs, sort of in the dark ages of targeted nuclease manipulation. We've done some things with TALENS and a little bit with the CRISPR system. So one thing we wanted to do was just to compare the efficiency of mutagenesis at different sites within the Drosophila genome. So these are the two sites that we've impacted. Zinc finger nucleases that I've talked about before, one in the rosy gene and one in the yellow gene. And this is just assaying the alleles of these genes one at a time by crossing the genuine mutants once the plasmid that have expressed the nucleases have grown up and can be evaluated genetically. So we've got, in this particular set of experiments, 20% of all of the alleles at this rosy site were mutated by non-homologous enzyme. There's no donor DNA in these experiments. And at this site, in the yellow locus, the number is about 5%. So we tried TALENS for these same sites. And as closely as possible, we overlap the recognition sequences for these different platforms. So the TALENS at this particular site in rosy were effective, but not as effective as the CFNs. They're quite effective down here where we never made CFNs, so we couldn't make a comparison. At the yellow gene, the TALENS were very effective at a site down here, and we got no mutants out of something like 100 alleles tested. So I don't know what the limit of, if we assayed many more alleles, we could find a few, but at least to the level that we tried, less than 1% mutagenesis here, even at a site that overlaps the site of the CFNs. When we tried CRISPRs for these same sites, they were kind of disappointed at rosy, but wonderful at yellow. So, first lesson here is you can't always predict who the winner is going to be, which platform is going to behave better. Another thing to say is that I doubt that the difference here is due to chromatin structure or something epigenetic going on, because we really tried hard to overlap the recognition spots. There may be some flaws in the design of the binding domains for any of these nucleases that we just aren't smart enough to recognize. It is true with the CFNs that we picked sites that were made up entirely of GNN triplets, and those are triplets for which there are the best fingers available. So we did bias toward good sites for the CFNs, but as far as we could tell just by inspection, there wasn't any reason to suspect that the TALENS or the CRISPRs would behave better or worse at any of these sites. And just before I go on, if you have only one gene to target, and you want to target it over and over and over again, for example, if you're doing human gene therapy for a particular disease and you're always targeting the same site, it doesn't matter which of these platforms you use, because as long as you get one good reagent, you can use that repeatedly. So for general laboratory use, people have jumped on the CRISPRs for reasons that are illustrated here. They're easy to design, they're effective in general. But if you have a single target, or just a very few targets, you may want to think about other aspects of these nucleases. Jin-Soo Kim put up a table that showed some of the comparative features of the nucleases. And if you can devise something that's really good for your one or a few desired targets, you can go with that regardless of what the platform is. We also looked at what types of mutations are made by these different nucleases. We have lots of examples, just sequenced examples from ZFN at a limited number of targets on the way. But what we find is about half of the new mutations are simple deletions, and there are a lot of new mutations that have insertions of some sequence. The TALENs and CRISPRs tend to, in our hands, in Drosophila, yield a much higher proportion of simple deletions with only a few insertions. And I was quite interested to see Prashant Mali's slide showing that different targets may give you a different balance of insertion or simple deletions. And that's true at some level for us, but not quite as dramatic as it can show. It's also true that the deletions are bigger. This is, again, a limited number of targets, but with TALENs, and a quick survey of the literature shows that this is true too. Amongst the simple deletions, the TALENs make bigger deletions, like small deletions with the ZFNs. But we're still talking about a relatively small number of base pairs. Just to show how little we genuinely understand about repair pathways, I want to describe this one mutation that we picked up. So sometimes the insertions at the target site are large enough that you can identify their source. And sometimes we see genomic sequences that we can identify from other sites in the genome that we picked up at the cut site. This is one that's very bizarre. It's a 720 base pair insertion into the rosy locus after the completed. 704 of those base pairs corresponding with one base pair mismatch to the archive genomic version of this gene called Gilbert. But they aren't contiguous 704 base pair sequences. They correspond to a part of exon 1 fused exactly with part of exon 2. So the intron is cleanly missing in this insertion. And I don't see any way around the conclusion that RNA, either directly or indirectly, provided the template for this insertion. The other thing is that 9 base pairs at this end and 7 base pairs at this end correspond to neither the original target nor Gilgamesh. So they came from outer space. My favorite hypothesis is that there are often untemplated additions at the break site before things are ultimately completed. And I wish I had a good way to test that. There are lots of different types of events that are stimulated by cleavage, and the frequency of the various outcomes, as I've emphasized, depends on the biology of the system. How many systems? Well, there are well over 50 different organisms throughout phylogeny that have been successfully targeted at genomic loci with these various types of nuclease. And this is as of a couple of weeks ago, and I'm afraid to go look, see how many new ones have appeared just recently. And I did something to infuse this. Okay, now I'm just going to go quickly over a few applications that have been made that help us to look into the future. So this is a study that came out of Dan Wojtas's group and the Selectus Plant Biology group in Minnesota, where they used TALENs to mutate genes that are responsible for biosynthesis of various oils in soybean to try to make soybeans that give you puffier oil. This is one that I'm pretty familiar with. Scott Fahrenkrug and colleagues have begun working on at the University of Minnesota and a company called Recombination. And this is to genetically dehorn cow. So most dairy breeds have horns, and the farmers saw off the horns to prevent the cows from injuring the farmer or each other. There's some natural beef breeds that don't have horns because of the mutation that's been identified called pole. There are actually at least 3 natural independent pole alleles. And what recombinetics proposes to do is to use TALENS to introduce the whole mutation into dairy breeds in a genomic background that's favorable for milk production. And you could do this by crossing beef cattle to dairy cattle and then back crossing laboriously to try to get the favorable dairy background again, but you still would have some remnants of the beef gene there. This is a very, very clean way to do it. This and the previous are a couple of agricultural applications. Thinking about sort of the broad range and focusing on human therapy, people have done a number of clever things to try to adapt these systems to human gene therapy. This is one that comes from a collaboration between the laboratory of Cas RNA-guided and Sangamo, who provided the ZFNs. So in hemophilia, where the mutations are in factor 9, the mutations are spread throughout essentially all the exons in the gene. And so to try to correct all of these with individual nuclease cleavage would require a lot of different nucleases and a lot of different donor DNAs. So what they did instead was to produce a pair of ZFNs that will cut within the first intron, and then provide as a donor DNA essentially a partial cDNA that contains all of the downstream exons with a splice acceptor and a termination site between the homologies that will allow this to be homologously incorporated into the first intron. And so that's a way to get correction of all the downstream exons in a single blow. And because of the way this is set up, the pre-existing downstream exons are never even expressed. Another approach that I like quite a lot, led by Koske in Alan Bradley's lab, again using nucleases provided by Sangamo. What he did was he took cells from a patient who was suffering from al-antitrypsin deficiency due to a single base pair substitution that converted a glutamic acid to a lysine. And so he took fibroblasts from this patient and made iPS cells with very low efficiency, which we found on that, and then used ZFN to cut very, very close to the mutation site, and a donor DNA that has the wild type sequence, along with a selectable marker. And if efficiencies were high enough, you wouldn't need the selectable marker. This got him a way to actually find the cells that had been cut and incorporated the donor. But the marker was within a piggyback transposon. So when the piggyback transposase was expressed, the marker was excised, and piggyback is one that excises without leaving a footprint. So there was nothing left here. There was a little bit of recoding right by the site of the mutation, but the only coding change after the donor incorporation and the marker decision was this conversion back to a glutamic acid codon. And they showed that they could differentiate these cells into hepatocytes in vitro and wild type alpha-1 antitrypsin. They could transplant them into immunodeficient mice and get homing of these cells to the liver. And you can imagine that a third arrow might have gone back to the patient who provided these cells to see if you could repopulate his liver with whatever. So I think most of you are probably familiar with this gene therapy trial that's been going on for quite a number of years now, 6 or 7 years. Collaboration between Sangamo, that provided ZFNs for the CCR5 gene, and physicians at the University of Pennsylvania. And, just to go quickly over it, they took patients that already were HIV positive, harvested circulating T cells from them, and expressed zinc finger nucleases specific for the co-receptor HIV that's required for infection called CCR5. When they evaluated the CCR5 status in these cells, just roughly, they found about 30% of all the CCR5 alleles had been mutated by non-homologous enzyme called ZFN cleavage, and about 10% of the cells had biallelic knockout of CCR5. And when these were put back into the patients, they provided an HIV-resistant T-cell component to the immune system of these patients. And the results of that trial, although they've been presented at meetings for a long time, have only recently appeared in published literature. And as you might imagine, they're now trying to go to precursor or stem cells to provide longer-term fix HIV resistance for these patients. And if you think about how gene therapy might go in humans, the applications that have been done so far are modifications that can be done outside the body. So you can take stem cells, circulating cells in that one case, and maybe do this in stem cells, embryonic stem cells, or induced pluripotent stem cells like USA did, and modify them and then put them back into the patient. If you think about genuine in vivo gene modification, then delivery becomes a very serious issue, particularly for diseases that affect the nervous system quite broadly, multi-organ diseases like cystic fibrosis. How are you going to get the therapeutic reagents to the cells that are affected? So you begin to think about germline gene therapy. That's what we do with model organisms. We do the modifications in the germline so that we can study this in the immediate and subsequent generations. And germline gene therapy is sort of scary to think about, but the tools are all there. And I don't know when the first of these experiments will be done, but I imagine we'll go on. So that's all I have to say. I want to thank the people in my lab who contributed the most to this. Of these people, only John Trackman, my technician, is still with me. Kelly and Marina were postdocs in the lab. These three folks were graduate students. Dave Siegel has his own lab at the University of California, Davis, and is working on therapeutic applications of these nucleases. And I want to thank NIH for support. Early on in this business, for me, it was very difficult to get NIH to give me any money because no one believed it would work. So we're a little bit lucky that it did work, and getting funding was a little easier. And I've had a lot of collaborators over the years, people in my lab, and then people we've collaborated with to apply the technology to a variety of different organisms and systems. It's been really fun for me to learn about the different systems that other people work on. And to see how the technology is growing and is visiting. And just in the back, so this is the University of Utah campus, the medical school where I'm located, it's just up here. And this is just a sort of typical day in Utah, showing the mountains where the ski resorts are located, just up a couple of canyons back here, roughly half an hour from Montana. So if anybody comes to ski in Utah, come visit me. We can talk a little science between runs down the hill. So thank you very much. I'm happy to do that. Yeah, that's what I meant when I said there was nothing obvious in the designs of the TALENs or the CRISPRs that would have made us think that wouldn't work as well in the case of the data. So we need, as you say, we need a training set, and you know, the extent to which that will be simple, if the design principles will be simple, we don't really know either. Maybe some complexity. But it could still be that there is some influence in the chromosome that particular sites, one platform or another, might be more sensitive to what's going on, either the nucleus or the location or
Another question I have is about the RNA as a candidate for repair. Is the RNA that particular one very highly expressed?
I don't think Gilgamesh RNA is particularly highly expressed. One of the difficulties we have with some of these experiments, and it's true for all of them, is that the outcome that we ultimately score doesn't happen within 2 seconds or minutes. We're scoring hours or days down the road. So we don't honestly know exactly when the events that we see are happening. So I do know that that particular insertion happened in larvae. This is at a time when we were doing e-shot for expression of the ZFN. So it happened somewhere during larval development. It happened in the germline because we only assess the germline by process. And I don't know, frankly, what the level of Gilgamesh is in germline, but I don't think it's highly expressed. We do have one insertion like this that came from histone locus and one that came from the ribosomal locus. They don't cross introns. I mean, they're from regions of those genes that don't have introns in them. So we don't know whether they're from RNA or from genomic DNA. But those, again, those are highly expressed RNAs. Histone message and ribosomal RNA could be that they were RNA from the
Curious if you could comment generally on where you see the field moving with respect to how to improve HR over HGG. Are you see people using knocking down factors or fusing other HR-related factors to some of these nucleases?
Right, so I have some ideas. Transient knockdown of non-violence engine seems like a good way to go. There's a published inhibitor in the United States. It has a rather poor affinity, but it's pretty high concentration. And I haven't seen any follow-up on the original study wasn't done with gene targeting particularly. I haven't seen follow-up studies with bias to the nucleus against primary. So that's a possibility. Elevating HR, we don't know what's limited. So, RAD52 has a little impact in the mammalian cell situation for people. If you knock it out, it doesn't make much difference in try to overexpress it. It doesn't make much difference. RAD51, I don't know how much people have actually done with that. Again, just overexpressing a single component may not be adequate. If you haven't chosen to eliminate one or several factors together. So, it may take, I think we were talking before about, combining factors, lots of candidates, trying to do it together the way the al-antitrypsin factors were discovered, and then winnowing away ones that are important. I don't know anybody really has the energy to try that. Another idea that I think I'm going to try when I have a chance is to try to introduce recombination factors from simpler systems and see if they will take care of it.
The donor DNA, is that saturated in the, I don't know to what extent concentration of that would increase the concentration of improving it to the ZFN
So, in situations where it's been looked at that I know about, the donor DNA concentration itself is not relevant. And one of the really surprising things to me was that the initial experiments we did in Drosophila, homologous recombination was mediated by one copy in each nucleus of the donor DNA that was liberated from a topic site by ISCP nucleus. So one copy of the donor DNA, not 2, not 100, 1, and yet the homologous recombination is quite effective. And when people have done experiments in typical types of homologous recombination, they find that the search for homology is almost never limited. And in fact, the way we all now make Pecky type targeting work better is not by increasing the concentration in the donor, but by making the target interesting.
So what about, so you mentioned the topic set, there was a side of access to one. Is the excision, or does it depend on the way?
Yeah, so we actually did that experiment where we have an integrated copy of the donor. We can actually, it's a two-step preparation. Flip excises into the circle, highest one next to the middle. As an integrated donor, the efficiency of utilization of the target is quite low. As an excised circle, it's higher, and as a linearized excision product, it's higher still. And we didn't do that over and over again in different backgrounds, but that was the observation. Published in 2003.
And can you comment a little bit on the possibilities of incorporating the various components required for the independent into one internal line or internal lines across the that seems to be given higher efficiency than the number of them, but also there's complexity when you go to a fix. On that potential ways of figuring out any of the proportions.
Yeah, so in a number of organisms, people have tried to set up Cas9 in the background, integrated Cas9, either a decision promoter, or a germline-specific promoter, or a tissue-specific promoter. But you keep changing your mind about the targets, so the guide and the donor are different from experiment to experiment. And at least in Drosophila, you'd rather not have to integrate them to get them to work. So people, I think, are mostly using energy connection to get the novel guide and the novel donor in. But it's certainly possible.